My name is Oliver Fien. I am director of the West Coast Metabolomics Center here at University of California in Davis. We are very pleased that so many people join today's monthly seminar that is given by Professor Jeff Chia. He is associate professor in large data analysis at McGill University in Canada. He's a Canada Research Chair Tier 2 in Bioinformatics and Big Data Analytics. And he's also cross-appointed with the Institute of Parasitology. His age factor is 45 with an I-10 factor of 69 and over 25,000 citations, with 5,600 citations in last year alone. He has a Bachelor of Medicine from Beijing University Health Science Center, and then a master's from University of Alberta. So he did his bachelor in 2001 and moved to Canada in 2004 and got his PhD in 2011 also at the University of Alberta. From 2012 to 2014, he did his postdoctoral training in the research area of next generation sequencing and systems biology at the University of British Columbia in Canada, and joined McGill University as an assistant professor in 2015 and became an associate professor in 2020. His lab focuses on leveraging bioinformatics, metabolomics, and systems biology to study the effects of microbiome and helminths and environmental factors on health and disease. He has multiple awards and recognitions, including, for example, the Global Highly Cited Researchers, but also McGill's Principals Prize for Outstanding Emerging Researchers. And today we are pleased to welcome him for his talk on from raw spectra to function insights and Multiomics Integration, a deep dive into MetaboAnalyst 5.0. So that's the latest version of MetaboAnalyst. For people who want to know more about MetaboAnalyst and want to learn more about MetaboAnalyst, Professor Gia also graciously accepted our invitation to be an instructor in our Bits and Bytes courses series. So he will instruct this on October 6th. And you can already register for October 6th on our website. And there he will do exercises and really training on MetaboAnalyst. All right, so without further ado, I give the microphone virtually to Professor Chia. Thank you for coming.
Oh, thank you, Dr. Fan. It's such a great pleasure. Oh, hello, everyone. It's definitely my great pleasure to present this topic to this audience. And so here's background Dr. Fan already mentioned. So my background actually quite diverse, from medicine, immunology, and then my PhD is more on metabolomics, bioinformatics, and postdoc is more RNASeq, next generation. And actually, I think my more deep into metabolomics in the last few years, because I was able to acquire a Q executive, basically a mass spec in my lab. So I actually go deep into the metabolomics. Before, it's more from a data science point of view. And I was able to do the computational and do some analytical. And we also have a C. elegans in the lab doing study aging and host microbiome. So today's topic is mainly focused on MetaboAnalyst 5.0, because that's people are interested, but I will briefly touch on other research area because it's all seems big pictures related. So here's a thank you note for my team and for the funding agency. So there's brief period last about in late July or August, something we think is promising and they're allowed to have some gathering. So we both wear masks, but for the photo, we don't want to wear it. So quickly took the photo. But now it seems quite remote. So is on first is my general understanding about Omix data analysis, and then we focus on metabolomics. And the overthinking is that metabolomics is not special. Okay, it's part of the omics, and we can benefit a lot by learning from other omics. And there are certain things metabolomics is more or less have certain unique the characteristics. We need to pay attention to that. But if we learn the overall patterns, we can easily do multiomics and finally, I would like to touch a bit of the ongoing effort and other tools. And I noticed this is a very diverse audience, so some of the tools may be useful, even I don't talk too much. And this is one actually motivated me to pursue my PhD. I was a medical doctor before, and treating patients one by one is satisfying, but it's not as satisfying. You hear big data, you touch a lot of the potential applications, and you opening your mind. So my time doing PhD is more microarray, but after that, then it's more next generation sequencing, and more recently it's a mass spec, high resolution, high throughput mass spec. So from the high throughput technology, go to omics data, and finally we want to have an insight, knowledge, and therapies. There's a long way to go, and once the data investment is there, and we always want to push for the translation. That's the final goal. But it's not really one lab can do it. It's all about engage everybody. But if the omics data itself became a bottleneck, then we cannot engage everybody, because there's only a few people, only small groups can do this large data analysis or processing. So one of my passion at graduate discovery is actually I want to bring the omics to be more accessible, at least computational wise. So this gradually bring to the overall understanding. Here we have the genomics, transcriptomics, proteomics, and it seems there's diverse of the omics, but overall there's only two omics, NGS omics and MS omics. So this became the more converged into these two platforms. There's other platforms, but this two is I feel is more widely used, probably going to dominate. And from this NGS omics to MS omics, we can see from the genome all the way to metabolome and to phenotype, and we're more close to functions. And this is one that everybody is doing these days, one multi-omics to understanding functions, the molecular phenotype underneath it. And if we accept all these things is all fine, now we need to really understand what the omics can tell us. And individual omics have individual tools and with different names. So if we don't pay too much attention, we can feel overwhelmed. But overall, it's more or less follow the same patterns. And these patterns will help us really transfer your knowledge from one tool to the next tools and all combine them together, because they really share the same core concept. And there's only three main steps. One is data pre-processing or data processing and the quality check. So this one is probably takes your most effort. Why? Is because here is more technology specific. At least we need to understand the next generation sequencing and MS if we really want to just focus on these two. And also, when we understand it, we will be able to do the quality check or quality control. And garbage in and garbage out. So this part is really, you spend almost half of your time to make sure with the time you are going to invest, and discovery will be valid. And if we can get over this stage, and all the remaining stage will be more or less shared and will be transferable almost. And it is statistical analysis and visualization. And what this tool try to do is once compare, for example, t-test and ANOVA, this is just compare individual features or genes or metabolites is all fine. We do clustering and we do classification. And for example, we do PLSD, random forest is all fine. So all of this is more or less used across our omics. And the last bit is interpretation, like functional interpretation. This one is all shared, more or less the same concept. And enrichment analysis, pathway analysis, although the library will be different, actually the library quality will determine how trustworthy is your data. So, but compare or create a library is not that high tech. So we just need to do it carefully and make sure it's accurate. And overall, in data analysis across omics is really just this high level should be shared across all the omics. So, so this is the data analysis. So it's easy to see in the concept, but when we actually need to do it or implementing it for the developers, and there's tremendous challenges. A lot of tools being developed, not being used or not being maintained. It's because this challenge is not being well thought out at the beginning. So in the end, it's become hard to maintain. The data first for the omics data is very big, and especially raw data. And download, upload, and process all takes tremendous resources. And maybe in the future it will be easier, but currently is that most practice is bring the data to analysis. Is for example, the below is really you use a process data, a data table to do analysis. But the raw data itself usually processed locally. If you do a genome center, metabolomics center, advantage is that they process the raw data for you, and you get a concentration table from metabolites or gene abundance table. And because the raw data is big and too hard to move around, so usually done locally, they follow standardized operation protocols. And for process data, and this is your job now, and you can use different tools to upload to like web or cloud to do data analysis. So overall, we have a very clear conceptual distinction between when to put the data to the cloud, when to do it locally. So when we step in this way, and much easier to maintain. And this is another thing is for the developers, like this for me, it's at the beginning it's so hard to address everything, but when we separate the concerns and became easier. So the concept for doing this, all put them together, is that we have the local raw data processing, like a genome center, metabolomics center, and we have public cloud. And the public cloud will do a lot of statistical analysis. This is what how we really maintain, update, and for my group, we use Google, and it seems quite convenient and easy to do it. And we can put more stats and machine learning there. But on the other hand, there's a web browser, and your laptop web browser is so powerful. We found it was so useful. We spend more effort to actually put a lot of computing and down to your computer, and you can interact with them. And maybe someday it will be do it offline, and the majority function can still function in your browser. And in the end, is we want to engage you. We want to engage users. We want to make you feel comfortable and you feel you understand. So the goals, everything prepared is just for the final user. And how we make the life easier, how you come comfortable with interactions or analysis, we are willing to accommodate, willing to do it. So this is through the years, we want to learn what is the best patterns to make you start thinking, and rather than worry about the next step. So this is something is the user factor is so critical nowadays, because we cannot just give this is right, this is wrong. And a lot of times we don't know. We just give you the possible and allow you to further ask questions to get more. So this is something is that the overall design concept is we try to make things easier, but it's left to you to decide what to do next. And overall is with this thinking, gradually actually found it people like this more accommodative in environment. They start using it and asking questions, and also it can grow almost throughout the years. And this kind of the stats on the right outdated. I'm pretty sure now it's probably 4 million jobs, and continue to increase. I didn't use checking it anymore because by regulation we have to allow you to turn off cookies, so it's not that accurate. So this tool is we have developed for gene expression for microbiome studies, and this is for metabolomics. Have other tools, but all of them seems being well used. So I would like to just share some other tools. So while we actively and also we have something in our mind is what's the future, because you know, people doing researches and they use cell phones and doing stuff. It seems all very useful and sometimes enjoyable to use all these Facebooks, everything for the entertainment. But while we do research, we have to suffer from some interface that looks very unprofessional. So, and sometimes it's really make you don't want to continue using it. But we should go with the science. We should go with the technology. We want to go to the future. So what's the future? Is that we have tools, and currently what we are doing is more multi-omics, like OmixNet, Omix Analyst, and we also introduced more features for like MetabOmics and other tools for multi-omics if it's really relevant there. The future, at least, seems unstoppable by all these big tech, big companies, seems AI and VR. So we are already actively prepare for the future. So for example, Omix Analyst, OmixNet, you see it's already have the 3D component. Why is that? It's one of the requirements if you want to go to virtual. And this is something as we want to see when the best goggles or the VR equipment will allow us to go there, analyze, and while we walk, don't bound to a desktop. And this on the left side is the things we relate to raw data processing, and we are actively developing that. The thing that if we don't, if we cannot get a raw data consistently ordered to the ways meet certain quality, all the remaining will be hard. So this is something we start doing. Also move to the next one. Also we go back to the developing some raw data processing in C++. Our main tools we usually mainly Java and R, but overall, for the raw data, we need to go to use a different language, and we are developing some of the NGS and MS tools. So let's go back to our MetabOmics analyst. So one is I would like to share that was the motivation a few almost 10 years ago, is that I really drawn to metabolomics because it's just making sense. I'm reading papers from Dr. Fan published like 2001 that really inspired me to, okay, now no gene expression, we want to do metabolomics. That really speaks a lot of functions and phenotypes. And so I started doing metabolomics. Then I realized a lot there yet. And so a lot of people want to do it, but in order to do it is that you need to do statistical analysis and normalization at the beginning. Is even a simple PCA PLSDA is not there, and you need to buy some tool softwares which is not actually designed for metabolomics. And so eventually I said, okay, we need to do metabolomics. We need to develop the tools for metabolomics, just like what the gene expression microarray did for their tools. So that was a simple. So I started to teach myself about how to do that. And that was back in 2009, and now it's almost more than 10 years. And we have version 1 is released 2009, and 2012 is version 2, 2003, every 3 years we have a new version released. So the first 3 version is more for the generic omics. This is really, I'm just saying it's a feature table. And it's not that particular to metabolomics. So a lot of people actually use omics data or even gene expression data upload to do the statistical analysis because it is a feature table. And you can actually trick the system to allow you to do a PCA or ANOVA or something, heat map stuff. You can do that. It's only last 2 versions that became more MS omics. One reason is I have this arbitrage in my lab, so I can study it further, understand it, make sure I understand it better and feel comfortable before I can program things around. So today's focus will really on this more MS specific things. More generic, we have a lot of tutorials, and just you should be able to follow and also ask me questions. And so this is, I hope, is more solved or more accepted. It's just the recent features became more heavy, probably more specific to MS. So here is the overall flow if we do the LC high resolution metabolomics or on-target metabolomics. Is we need to do spectral collection. It's about 20 20 minutes per spectral. This is sequential. We are talking about LCMS, and I know probably other GCO will be faster maybe. And after we collect the spectra, we need to do spectral processing and compound annotation or identification and functional interpretation and integration with other omics. So this is a very natural path for everyone. If you want to do it, move to next. So the MetaboAnalyst version 5.0 is actually just driven by this whole process. We want to smooth every steps. So you just click your button and thinking and move to next. So this is a whole design for MetaboAnalyst 5.0. Let me focus on the spectral two features. This is one of the biggest bottleneck for me when I want to do MS omics or metabolomics. So at the beginning is that we need to deal with the raw data. Okay, raw data processing is tremendously challenging, and I want to postpone it unless I have the machine. I can back and forth double check and see the features and double check we found the right ones. And so that's why I postponed only recently to do it. There's multiple tools, excellent, powerful tools. We use it a lot. XMS, OpenMS, and MS-DAOs, and MZMine3. We use it's not just we develop tools, only use our tools, but we use all of them and at a certain time. And but we also borrow a lot from their concept by benchmarking against the different tools and then see what's the difference and learn from that. So for web-based tools, we cannot allow too much interactions, even it's possible, but when you have a very long sessions and back and forth, it is just not efficient. Also, the bandwidth will not allow, can be interrupted. Our goals for developing the raw data, raw spectral processing, we want to be automated. So you submit the data and have some minimal setting of the parameters, and the system will take care of the remaining. And this is just like Tesla driving. I cannot help myself use this image because I'm always thinking that when I design and implement. So the overall thing is that can we make it more automated and learning from like best practices. So when for spectral processing, there's too many parameters to tune to get a good result. And the other one is functional analysis is very time consuming. And how can we make it automated? And this is one is not only do it, also do it fast. Because if we don't do it fast, and we cannot sustain. Basically, we are going to kill ourselves. If user submit and takes 2 days or 2 weeks, and the next user submit, they will snowballing. So overall, that we want a solution that is reasonable, that is fast, and it must be better than if you use default parameter. So how can we go about solve these issues? And one key observation is that MS spectrum is quite sparse, and this is interesting. But it's when I see this, I said, okay, why we don't focus on the places that more have the signals to train rather than using the whole spectrum, which is very big, but it's signal. When we started thinking about that, we found it's more close to coding regions in genome. Like say, we only have about 5% of genome have the coding regions. At least at the beginning, we know non-coding region probably we call it garbage. Now it is have functions, but at the beginning, we want to focus on things we feel most likely give us the knowledge or things we want. So this one is the driver of our design. So we want to use this coding region to optimize parameters and not using the whole spectrum. And in that case, we feel this region contains more real peaks, at least enriched for real peaks. And actually in the long term, we actually can go to the spectrum and find a specific MZ values. And based on what we want to question this whole pathways, we can actually tell where to look these regions and just train on these regions, not use the whole spectrum. So there's some issues with that because your access spectrum is not that easy, but now we overcome that. But at the beginning is that we cannot do that accurately. So we use actually chop the spectra into different bins and found those with the high abundance signals. So this is a current implementation, but we know we can do a much fine control nowadays. So the idea is we found a region of interest or the coding regions. So we don't use the whole data. We only use the pooled QC samples. For example, if you use 100 samples, like 10 QCs, we probably just use 8 QCs or 5 QCs. As long as they are more or less similar, we just use them to train. Or if you don't have QC, and the next best one probably use the more largest files, but we strongly recommend the QCs. It's just if I don't allow QC, and if don't allow people to proceed, and they will not happy. So in the end, we said, okay, you use the largest files, top 5, top 10, and then we found the regions. And these regions, some people are afraid that, oh, you're probably going to lose a lot of low intensity signals. When you actually zoom in, you will see a lot of signals, low intensity, high intensity, all there. So it's probably have some bias, but overall that it's really not just a high abundant peaks. It's when we have a rough region, and within this rough region, there's a lot of the variety of signals representative of the spectrum. We continue benchmarking and till now. So we published actually early last year, basically it's 12 or 18 months ago. And we benchmark against the IPO. So IPO clearly is the best one, but it's running for 2 weeks, and ours is running for 20 minutes and or half an hour. And we are second best, but we are talking about from days to minutes. And in terms of linear peaks, we use dilution series, and we can see the MetaboAnalysis, which is the tool algorithm. We have more linear peaks, and this is called reliability index. So overall, that we think it's doing a good job. Okay, from faster and more linear peaks. And just 2 months ago, and we did another case study. It's using malaria data. It's about 200 samples. We just use default QE. The default is really based on XMS Online, Q Executive default. And we found the peak numbers like this. And we use auto-optimize like and but we really look into the details of the peaks that annotated with isotopes ADAX. We found almost half of the auto-optimized is you can annotate. And with molecular formula assigned, it's more. And we can assign more putative compounds, and PCA have more variations explained. If we think our design of the experiment is capture the main changes in the stuff, and the PCA, the first 2 components, should explain more. So far, this looks meaningful. And we also have more significant peaks overall. So this not necessarily means right, but overall you feel that your design capture more biological meanings. So this is our second study. We actually did more studies with IBD earlier during our publication. Overall, that definitely better than the default, and much better. And with actually more cost, is about half an hour to 40 minutes. And parameter need to be tuned. They need to spend that additional time, but it's not too long to tolerate. So I know maybe some people can further tune the parameter better, which is totally fine. So we can tune, and then we show the parameter, and people can further tune from there. So I think this is a collaborative between man and the machine to move to next. So while we were doing that, and we continue developing, and we see this public all on nature method, we are more or less they have more or less followed a similar thinking, at least share some things. I clearly get what they try to achieve. And overall, that the thinking is here. I want the field expert to continue digitalizing their knowledge. So a lot of rules when you become making into open source tools, and these rules can gradually can really replicate or being enhanced by other tools, gradually become more accurate, because a lot of the things when we discuss, it is clear, but when you're actually doing it, it's very hard for a lot of people. But when we digitalize, make into tools, and people click button, get that expertise, that will be the way to go, and faster into the engagement and high quality data analysis. So for my group, we want to enable faster targeted spectral access. And basically, I'm talking about if we give a list of the MZ values, can we directly extract it from the spectrum? Not we cannot, we want to do it very fast. We're talking about seconds and through the hundreds of spectra. So this one will really require low level things, but it seems it's doable. We have tested, it seems fine. And the other one is integrating with no knowledge. So these two publications, they seems very, very promising. And the other one is if we can integrate with directly not networks of the reaction stuff, we can further improve, which is currently we are actually testing the approach on top of these approaches. And it seems that we can get better result, but it is not finalized yet. We are still actively testing them. So what I just described is on the underlying concept, what we have done. But for general users, you need to use the web-based tools. And if you want to use our package, it is OptiSMS on GitHub. We're active development. We're not that because we want to continue benchmarking and improve it. So if we click the raw spectra processing, and we go to this data upload, and you can upload about 200 spectras, and if you're a guest user, so you don't need to register, do anything. So you just click upload, and it's as a zip file. So you need to use the QCs, and because the QCs are very important for the parameter optimization. So after uploading data, which you have to do yourself, that's the only thing. But after that, you can just click this button, auto optimize. It will basically all these parameters will be disabled. That means you are not going to touch it, and the parameter will be determined by our algorithm. So if you choose that, of course, you can manually do it. We receive a lot of requests why they don't get a result. And we realize their manual parameters, not the errors. We don't even program to catch that. So we don't know, but then gradually realize there's some issues. We can let users know this is errors. You should readjust the parameter. And so the best way actually is try to use auto, and which is most time we'll go through it because it's try their best to find the parameters that making sense. So if you choose the parameters and click submit your job, and here's the things, it will start working. And you basically after 1 minute, so create a bookmark URL, you can save to your browser and or save to somewhere you can revisit later. Usually each job will take about 2 hours, 2 to 4 hours. So you should get it the next day. We have processed about 6,000 jobs. So it seems more or less okay. After that, you don't want to just download the result. You should continue to do the data analysis. So you click when you're finished, you click this page. We see the PCA overview, and this is the features, this is samples. This is not too big, but still a lot of features. And you can click actually see which features different or what's the EIC. So this features is below, and you can view these features And importantly, is every feature, every sample is clickable. You should be able to show the raw spectral EICs, what it looks like, and create something if you're really an expert in this one, if it's a real or not. And they do try to do some annotation, but we are not proud of whether that's the best. So this is something we just do the default annotation and then use the camera, but we want to improve it further with the new approaches to improve the annotations further. So that was the first one, how we do it from raw spectra to features. And then, so that concluded the first. Second topic is how we do it from the features, pick features to functions. So overall, that we have a list of features, could be a feature table, or just ranked features based on the p-values or t-scores if you do a t-test or ANOVA. And you want to understand the functions. So this is a lot of goals. And so we want to see which pathways changes. And from the path, we want to see which compounds is most meaningful, could be targeted by some drugs or therapies. And how we detect the function changes. And this is very different from doing individual data analysis or feature analysis doing the t-test ANOVA. And one of the biggest understanding is that functions are group behaviors, and we should be able to test the group behaviors rather than individual features. So the concept breakthrough in I think in 2006 is called one approach called gene set enrichment analysis. And what they try to do is they want to test the members involved in a function have more consistent changes. And for example, most move in one direction rather than random directions compared to the random changes. So this is a consistency defined functions. So how can we do that? There's a approach is called enrichment analysis, like over representation analysis, and the other one gene set enrichment analysis. And this is require complete ranked gene list. And this one is only need most significant genes and compounds. So this one is more or less testing, seems to be the same thing, but it do have different implications. So if we do the representation analysis, you just give the significant genes, the background is assumed. So basically the machine, the algorithm will assume certain background to do the enrichment analysis. So for microarray or gene expression analysis, this is not a big deal because the transcriptome almost comprehensively or completely. And but this could be an issue for metabolomics because we can only measure small portions of the metabolome. Also, different platforms have different bias. So this one is, although it's easy to use and a lot of people using it because you only upload the metabolites that's significant. But if you do this, a lot of time it could be biased towards your instrument, okay, your extraction protocols. At the moment, it's a more unbiased, more strict approach is actually more use a complete list, and the background is actually being generated by shuffling, by permutation. So it's the null background is from your data, from your which is defined by your machine, by your platform. So this concept is being illustrated in 2013 by Dr. Lee. So what he gave a name, it's called Multiomics. So in reality, it is I will say probably peak set. This is more fit what algorithm what it does. So overall is that he used. This is assumed to be a high resolution mass spec because high resolution naturally have a error to be incurred in the data. So based on that, we further do the putative annotation of the peaks. And this putative annotation peaks will contain the errors. But if we assume the error is random, we hope it is random, then okay, they will reveal the true functional changes. So even individual, there's errors, but as long as the error is random, they should collectively point to the right direction of the functional changes. So we know the functional or group level changes are robust to errors occur at individual features. So indeed, this one making sense, and when during validation seems working well. And so important thing that they don't need to do the accurate metabolite identification. They just do a fast putative comp. And so will contain errors, but it doesn't really matter too much on the end. But overall, if you continue to improve the annotation, the final result will be more accurate. So this does not prevent you from continue updating this annotation. But the most important thing is make the whole thing so fast. You can directly get to functions. So we have implemented both the GSEA gene set enrichment approach and the MicrobiomeMetabolomics approach in this MetaBioanalyst 5.0. So functional analysis, and you go to functional analysis. And when you can upload a peak list profile, this is ranked peak list from the t-test or t-scores. Or you can direct upload the peak intensity table from like XMS online or from whatever your local software. So if this been uploaded, and it basically it will go to the steps, and you just click, and they will have explanation steps, tell you what's the next, what's parameter. And you can choose either using the gene set enrichment GSE or using MicrobiomeMetabolomics. And I believe this one is from MicrobiomeMetabolomics pathway enrichment school. So it will tell you some pathways that being enriched or based on p-value, or some enrichment factor is how many compounds hit you expected based on random, but what you really see. For example, like a 10 compound pathway, you expect to see 2, but you actually see 9 compounds up regulated. So the enrichment factor will be higher. And again, it will be p-value will also be calculated appropriately. Overall, that this one summarizing the pathways that are more likely to be changed. And you can click each pathway to see the assigned compounds. And this is I think this CAG compounds, vitamin E metabolism. There's the tryptophan. There's something here. And will be from GSEA. One thing with GSEA is they can tell you both up-regulated and down-regulated. So like here, volcano plot, you can actually pick top here and top here, which is up-regulated, which is down-regulated. And again, is that you click, you will see that. And further down is that we projected the tools, the putatively assigned compounds into the CAG pathways. And you can click each node. If it's assigned, it will see what's the matches for this. In the future, you will see the EIC or XIC extracted from here, whether you think it is. So far that it is not related to the raw spectrum, because when you upload this peaks or peak table, and we don't have access to raw spectrum. Overall, in the future, it will link to that raw spectrum. The other one is if we do like say the t-test ANOVA, we found a significant ranked peaks, which is fine, but we are looking into the peaks that associated with the disease or with certain phenotype. Sometimes we just want to see some peaks that change together, co-expression or co-regulated together. And they are not necessarily have p-value regarding to the healthy disease or different things. They just seem to have a good patterns and seem quite interesting. And in order to do that, we need to engage users. And basically, the program cannot do it very well in a heat map. It select. So you see, you can apply different clustering, and you see some groups or peaks that have consistent patterns across 4 different groups. I forgot what is phenotypes. It's 4 different groups or 4 different cell types. It's probably dendritic cells, monocyte, or B cells, T cells. I forgot which stuff. Probably that's immune cell types. And if you see here, this patterns of metabolites that's very different. Could be a signal different cell types, and you can drag, use a mouse drag and select. It will be here. And we can actually test what the potential pathways underlying these patterns. And this is phenylalanine, lysine, purine, and this is seems like this. But we continue testing. Recently, use a higher resolution. Once we use a ICR, one of high resolution, we actually get much strong hit. So it's quite interesting to see the high resolution features and can show up here, and it seems all making sense. And so this one's really require user input. So you see the patterns, you want to find more, and you need to select and show this, then test the pathways. So I went through my second topic is how do we do from the features to the functions. Then next one is how do we from the functions to systems or multiomics. And what is the current thinking is that the multiomics integration is a very complex, and there's no consensus what's the best and what's not. Usually it's case by case. And most of the top publications, we can see a lot of the expert users, they're driven by them. They have certain designs, they have certain hypotheses, and they use their hypothesis to organize analysis to prove whether certain hypothesis is correct or not. So it's really have a strong targeted design approach, user-driven. So this part is really we need to learn and continue to learn and see whether there's certain patterns we can capture and put it in the tools user can use. So this one is really ongoing, but is most exciting part. And the other two part is knowledge-driven and the data or model-driven. This one is more or less, there's a lot of discussion, there's things going on, and we can actually implement some of them. And this is my next discussion. And one of the simplest is called pathway mapping. And so we measure the compounds, and we can actually measure, project them both to the pathways. Our naive thinking is that if some pathways changed, and both the genes and metabolites or protein metabolites, probably all of them will change. We hope so, assuming it's reach a stable state and not have some like temporal difference. I know there's a lot of the thinkings and stuff, but when we get a high level, we want to just see stable state, and there's a stable difference in the signature at both transcriptomics level or metabolomics level. So we can project them both to the pathways, like we call it pathway mapping. This requires you really have the compounds that been annotated or been identified. And this is mainly suitable for the targeted metabolomics. But what we want to do is a pick list. Can we do that? Because we can easily get a pick list. And this is one to latest version. And the other thing is when we can integrate, one is direct at the feature level, and the other one is at pathway level. So feature level is, for example, we combine the genes and metabolites. And the other ones, we only add their p-values, for example. The other ones, we test the pathways for metabolites. And for genes, we test, then we combine the pathway p-values, not feature p-values. So is that individual feature p-values, this is more like a meta-analysis, but we want to have the evidence significant p-value combined will be more significant. And overall, the pathway will be like that. The pathway levels, we test the whole overall pathway, and we combine them. Feature level integration is doable if we have targeted metabolomics, because we can have p-value for each compounds, then we combine with the significant genes. And the issue with this is we have hundreds of compounds, and transcriptomics have tens of thousands. So usually you're talking about 10 significant compounds versus hundreds of significant genes. If we directly combine them doing pathway analysis, the whole pathway will be driven by the significant genes. So this is something we noticed it. So a lot of people continue doing that, but we have to keep in mind the dominant by these genes because they just big, right? And in metaboanalysis, we do have people to have different ways to de-emphasize the genes and do it. The other one we have implemented is pathway level integration. This one is really clear. So we just do a pathway level p-values and integrate the pathways. And importantly, the pathway level allows us to integrate peaks directly because we can do a pathway analysis for the peaks just using MicrobiomeMetabolomics or GSEA. Then we do the gene set enrichment analysis at the peaks and we can integrate them. So this is we had implemented in 0. And meanwhile, we also noticed publications also last year, last 2 years, they're doing the same thing. So it's more or less we arrive at the same path. So overall, is that we maybe in the future we can borrow some subtle changes. Overall idea is that for multi-omics, pathway as a unit seems to be more clear and robust. And if we can do it, and that's the way at least easier to interpret, easier to implement. And this is one that shows in the MetaBanalysis 5.0 how we do integrating peaks with genes using joint pathway analysis. It is here. And so the idea, we have a quick tutorial is here. We have the peaks. If you're using peaks, it's complete peaks because we need to test the now background. So we need to do the shuffling to do the p-value calculation. And this is based on the MicrobiomeMetabolomics. And this one is you can use in the overrepresentation analysis or GSE, no problem. Now we use. Then we can integrate in pathway of metabolites, pathway of genes. And overall, we hope the signature is combined, they should have on this area. So here is that things, if we upload in this genes, and here's the on-target peak list, it will be a file because this is not going to copy and paste. It's a big file. And after that, you are going here, and this is arginine proline metabolism. This, I think, the immune cell, also the immune cell stuff. We don't see a joint signature. It's here. It seems both. It's detected by metabolomics and pathway only detected by transcriptomics. This is not surprising. Okay, this is quite common. So a little bit more on the multiomics is that metabolomics gradually moved to integrate with other omics, but there's other omics also want to integrate metabolomics. So there's always primary, secondary. So I found is if more developing of multiomics, it's better to put it in more dedicated tools. And we have the OmicsNet, which is for all multiomics integration, and this Omix Analyst, which is for data-driven integration. And we are actually updating these tools for 2.0, and we are updating it. So here is that we want to people upload the gene list, protein list, the transcription factors, the microRNAs, metabolites. And we added also actually taxonomy from microbiome, PICs, and SNPs. So these new types, if we want to bring them to the main knowledge of interaction, we need to infer them. So the PICs is infer, okay, or predict. And SNPs to genes, also there's predict. And taxonomy to metabolites, so which kind of the metabolites associated most signature of certain compounds. There's also ways to do that. And there's the overall that this is not direct, but it is computationally you can infer. Once you infer them, linking to this, there's a lot of how to say functional visualization to help you to develop the hypothesis. So this is something we are and testing more. And again, at this moment, we want to the visualization because when they get in multiomics, it became so complex. And if we hide too much details, and it will be misleading, could be something we don't know, and the user will naively believe. But if we want to bring out too much, it's overwhelming. And so it's always this happy middle ground, and we don't know what it is, and we're exploring, and sometimes use a. So for example, this is a three-dimensional network we have developed. You can do it using a browser. You don't need to install anything. And eventually, even it looks pretty, but it's really overwhelming. And our next step, we already implemented, is put it into a module. So from such complex things, we can actually put it into a different colorful bubbles. Each bubble is a module. And you can do an individual analysis visualization on the individual module. So the whole complexity of the network can be you attacking individual module one by one. So this is something we found we need to have a hierarchical approach rather than just everything and attacking everything. So it will be hard. So this is another one, Omix Analysis, we published last year to directly upload the data tables from different omics. We can combine them. Again, we want to emphasize there's a virtual reality theme here. It is not virtual reality yet because we don't have goggles, and but eventually, if technology allows, will be there. So you can see this is all the implementation in the 3D, and we have different multi-omics. We just interject them together and combine them, and using dimensionality reduction and visualization, we can actually see which compound drive the separation. Even the data point is so many. It looks a lot, but if we just see the patterns like different color bubbles and the main biomarkers of this loading plot, we will still see what's the main patterns, what's the main drivers. So this is my final slides. Hopefully, it's the last two slides. I'm not going to presenting my case study. So this one is about how we understand our data. This is omics data, metabolomics, transcriptomics. Then there's metadata, clinical metadata. There's published data, and there's knowledge, pathways, networks, and which more senior people probably have more knowledge. And overall, that we have domain knowledge, and the tools want to bring the visualization stats and help people to understand. And the key part actually is we need to develop better tools, intuitive tools. And on the other hand, it's about engaged researchers who will actually understanding and making discovery and move to next. So this part is quite an art. You need to explore different approaches to finally get there. So this is my thinking, and maybe in the near future we will get there. The Googles, Facebooks, they are really eager to jump there, but let's see. Also have some other data I'm not going to discuss. This is based on our recent COVID-19 data for biomarkers. I think I will skip that. And last two, one is microbiome and metabolomics. So I did a lot of microbiome metabolomics studies. And one thing we noticed that is that most study on the microbiome phylogenetic difference. But if we focus on the metabolic potential, metabolic distance using the GEM models or CAG, they are huge difference. So a lot of the predictions or assumptions using the microbiome phylogeny is way different from metabolics. So our group just did a very naive approach. We just predict the metabolic profiles directly using the GEM models, and we can get much better result. And compared to what the people usually do called pie crust, and we mirror in metabolomics, we see that actually the patterns hold. So the thing is that we need to develop tools directly from metabolomics. Don't use genomics stuff. They are quite misleading. So we hope they are consistent, but there's a huge difference. This is another one is we have done. It's called the Universal Platform for RNASeq and Annotation and Quantification. And this is my as Omix. Overall, I want to do RNASeq for any species, not related to just model species. And we have made some very good progress. And if people are in that field, and feel free to check it out. And we have got Omix Analyst. So thank you, everyone. Yeah.
Thank you, Professor Xie. That was a fascinating tour de force from raw data to functional interpretations. As usual, Dr. Arpana Vania from our center will now steer the discussion starting from the chat. Maybe Arpana focusing on like diversity of questions.
Yeah, we have a lot
Sure.
of questions. Thank you, Dr. Xie, for a great talk. It was really good to see such a big overview on the tool and what it can do in the future. So just to kind of kick it off, you had mentioned how many users you've had. I think the number was in the billions or millions. How do you handle 2,000 users per day? What is the underlying hardware that you use for MetabioAnalyst and all the other tools that come with MetabioAnalyst?
Yeah, that's 2,000. It's probably 5,000 per day, and we each use a lot of powerful users actually submitting a lot of jobs. So we see about 20,000 jobs analyzed per day. The trick is democratizing. It's using shared memory. So the overall is I didn't share the slides. I'm thinking when you have a tools for everybody to access, unless you tell them 1, 2, 3, click the same button, they will not do the same thing. When you randomize through the whole 24 hours, the CPU consumption can be really homogenized. If I'm using Google, I can check in how the memory being used. It's really not that huge. We're just using one Google virtual machine, about 64 gig of RAM. And of course, we profile our code very carefully. We want to make sure there's no memory leak, and any job will finish within 2 minutes. So there's no kind of if you have a job that's not being killed and sustained there, it will have a very bad consequence. But over the time, we actually did kill them. The trick is really be careful about the quality of the code and no memory leaking. And this is something, it's fine if you have a time doing that. The other ones that what we are learning, my team is using a microservice or Kubernetes. And so you can isolate different components, even one fails, and you can troubleshooting that. But overall is that when you have a scale, you have economy. So there's no much secret. It's just a software engineering principle. Yeah.
Great, thank you. I'm sure a lot of our programmers could appreciate that answer. There was one user and possible user that had asked, if you have more than 5,000 features in a data set, these features would be filtered by MetabioAnalyst. How are these features being filtered in theory or in practice, really?
Yeah, I get a lot of question on this. I'm happy to answer that. The thing is, cannot really take everything and analyzing everything. We need to focus on something that carry the big pictures, most signatures. And so if we detecting 5,000 features, usually at Omix, all Omix tools assumptions is that there's a small portion of the features that changed due to your perturbation. Like a small portion means almost no more than 25%. That means 75% maintain a homogeny. They shouldn't change that much. So this is all microarray, all other omics being designed, all the tools. So a lot of most famous Lima for the microarray gene expression, they borrowed a lot of the information across because they learn they are in a homogeny, they don't change. They can borrow the variance to improve the consumption, improve the variance estimation. A lot of the stats are really based on this assumption. So if you have 5,000 features, probably only 1,000-ish could be potential biomarkers. So if we filter out some of the feature and the tail, if you rank by the raw variance by the abundance, the the loss will be minimal, but the gain will be a lot. Why is the statistical test will be more powerful due to the multiple testing? And so, at least from data science point of view, I think it makes perfect sense to not allow people to analyze everything, especially statistical models that goes in as a normal distribution. If you including all this kind of at the tail, a lot of program may not working well, misleading. So this part is that I'm debating with myself, but I need to be convinced by some good publication in a statistical journal. And other than that, I think that's something I would like to enforce good practice by filtering out some like 20% or maybe half. Sometime if you have a lot, 10,000 features, maybe half will be gone.
Yeah, I think that's an important note to make. You know, it's important to filter out anything that's not, you know, useful for your statistical analysis, but then also try to capture as much information as possible. So yeah. I could see that being kind of a dilemma. There is a question here, and it's applicable to not only new users to MetaboAnalyst, but those who are getting started in metabolomics. What kind of raw data do we input into MetaboAnalyst 5.0 platform for metabolomics analysis? So what is the input for this tool?
we supporting all kinds of tools, all kinds of input, like as just CSV files. I know people want me to support in the Excel spreadsheet, but overall that just a data table of the compound concentrations or peak intensity tables, you save as CSV and upload it. And you can also have a list of the peaks or list of compounds. So you feel free to explore. For the raw data, we only support something like MZML open source format, but I don't suggest this one for the beginners. Yeah.
Okay, great. Thank you. There's a question here related to over massaging data, you know, that can take place, of course. How can a user spot if their data is over massaged in MetaboAnalyst or in, you know, even in general? In other words, what would you suggest to QC the data processing step?
Yeah, that's a very good question. The only thing if guaranteed to work is that increase the data size. When you have more data size, you see the patterns are real or not. This is something cross-validation or stuff. Overall, is that when you have a smaller data and a lot of people start thinking a lot, overthink, and you see a 6 data point, you think there's a 3 and 3, there's 2 separations, then they write a paper about that. And yeah, I see clinical, they don't get a lot of samples, but they really want to have some findings or descriptions. I don't have too much to enhance. The only thing I can help is you not massage is analyze the data comprehensively and think what the information tell you. And the other one is public data. If you don't have a lot of data, what's the public data? If it really the previous data and more evidence need to be provided. If it's more or less consistent, then probably less. Overall is the current trend based on the statistical literature, p-value is not being that important. Overall is the comprehensive context and that's supporting whatever you want to say is important. So it's a whole package deal. And you can have small data, but you leveraging a lot of public data, or you have a smaller data, but your analysis is very, very rigorous and the same. So it's hard to give a single answer, but overall that live with your data and try to understand what the method tells you, and try to use different methods. Are they consistent or not? So it's really a dialogue between you and the tools and data and the literature.
Great. There's some questions here regarding the pathway analysis that can be done through MetaboAnalyst. Specifically, how do you treat biofluids? Metabolites and biofluids represents lots of different origins, not only cellular pathways. And is the pathway mapping based on KEGG pathways, or do you use other tools as well? So.
Yeah, these are good questions, and I think that one need to be updated. I think we have better understanding of the cellular component for different metabolites. It was done earlier based on HMDB 4.0 or 5.0. And yeah, that's one part. What's the next question is other pathways except the KEGG. So KEGG is free for academic. So okay, so I think it's more or less leverage that knowledge. It seems okay, and we are more involved with maybe other metabolic specific stuff. We now using a lot of using RECON 2 and RECON 3, and that's the overall in connectivity, but they don't have a very well-defined pathways. So this is quite a dilemma, and also something I think the field should address better. And I agree with this pathways. So we're using KEGG, and we also try to use others, but it's just we need to have a consistent, high quality, well agreed upon source for that. Yeah.
Great, thank you. The next question is on OmixNet, which was something new that I learned during your talk today. Does OmixNet use a mix Omix library? I think that's a library name. Models such as Diablo or Mint. And the question, the person that asked this question, they wonder if at some point users can integrate omics and non-omics data, such as complete blood count, to apply for, practical applications in different, settings, such as hospital daily protocols. So really it's, asking about like the future of MetaboAnalyst and where do you see it going?
Oh yeah, let me go back to the first questions. So I introduced the two tools. One is OmixNet, one is Omix Analyst. So OmixNet is just a really network. You have a list, try to connect with a known database, protein-protein interaction, microRNA to genes, and transcription factor genes. And metabolite is basically enzyme to metabolites or something reactions. So we just connecting the tools and allow to visualize. We do a little bit of computational imputation. For example, SNP, if you input SNP, you find the genes that are closest to it and try to link the SNP to genes and to the maybe pathways protein-protein interactions. So this is really just a visual linking and visualization. And if you're talking about Mint and Diablo, it is here. It's an Omix Analyst. You need to upload a complex, upload the data tables. It will use the dimensionality reduction and project. And Diablo is one of them. Actually, we have 12 different methods for dimensionality reduction. Okay, so Mint and Diablo are both there. So this is we try our best to make it more visually appealing, because that's a really multi-omics, multi-variate statistic is so hard, and we want to bring it to be more easily digestible, just like this. But this is this result probably from Diablo. So regarding a second one is actually we already addressed it, and hopefully we're starting to address it, but in a way. Let me see, and I update that hopefully. So MetaboAnalyst already have the capacity to deal with metadata from clinical. Let me see here. If you see this one, you see statistical analysis metadata table, and you upload your metabolomics table as a compound concentration of peak intensity. Your metadata table, you can it's also another table of clinical metadata. Like we have this one have an example is all COVID. We have the all clinical COVID data, and usually you have about 12 columns: gender, status, age, everything, high blood pressure, or the cytokine count, everything, and you can do a co-analysis together with the metabolomics. So this one, this this demand is so strong, and we cannot postpone MetaAnalysis 5.0. We added this after publication just because when we analyzing COVID-19, and we realized we have to have tools to support people to do this. So please try this, and you should be able to upload the gathering metabolomic data to the analysis. And there's some of the new statisticals to do the covariant adjustment and stuff. And I think this is a lot of clinicians that asked me. So I think it should be helpful.
Great, thank you. There's a question here. Do you have plans to provide MetaboAnalyst tools for QC to evaluate study or data quality, such as pooled QC results, blank results, dilution series results, really anything to evaluate a data set in a way that you have like visual aids such as networks and PCAs and things like that?
Yeah, well, thanks for that question. So I think there's another tool called NOREVA, Normalization Quality. So because that one is designed for the quality checking. Oh, I forgot what was the full name of the web-based tools. For MetaboAnalyst, it's possible. I don't know. It's because the next one is actually improve the annotation from peaks to compounds identification to be as better as possible. Before we use camera, but we see there's new publication. We want to continue doing it. And next support was MS. And we back to the QC stuff is really depending on the our resource, how many team members they can do it. Also depending on our requirement of the urgency, because there's a really our practical understanding in our day-to-day operations. If this became a huge issue within our team, then there'll be a big motivation to push it forward. At the moment, our pushing the next one is MS, to be honest. Yeah.
Okay, great. Thank you for that. There's a few more questions for new users. It seems like in the audience, we have a lot of, new users and folks that are new to metabolomics, which is great. Do you provide like a straightforward A to Z steps on how to use MetaboAnalyst for processing LCMS data?
Yeah, that's we would like to do it. We develop protocols to update once in a while. And for example, yeah, for the raw data processing stuff, we haven't do it yet. But yeah, it's on the plan. But for all other tools, statisticals, everything, we already have I think it's 120 pages protocol step by step to do that. But the spectral processing here is we are not getting that because we still continue doing the benchmarking ourselves. We're analyzing thousands of samples. We want to improve and benchmarking and improve. So once we feel it's very comfortable, we were going to lock it and basically spend more time doing this documentation. So far that it is available, a lot of people are already using this. And so overall, that if you use web-based tools, it will more or less to think too much. Just upload and click using auto-optimize. That's the whole point of simplified.
Yeah, and from personal experience, using the web-based tool, there's a lot of information to guide a new user. So, whether it's like test template data sets in terms of formatting and all of that. Dr. Xie, is there a discussion forum or a Slack, that for users, to get these kinds of questions answered about format and, what parameters to use, things like that? Yeah,
it's a good question. And there are two things we want to do, but delayed by the COVID or stuff is one is we want to develop in a forum, indeed dedicated to the metabolomics, multiomics, and especially for my team. And the forum are going to come with a chatbot. The chatbot, we already pre-trained them to answer a lot of questions. I also want the chatbot to be learned from expert answer. So basically, the forum will to answer other people's, and our team will be doing it. Other people can freely answer the questions, so the whole community can share the resource. And we hope to train maybe a chatbot to learn and help new people to directly, what's your question, and clarify, and just charge them to the right forum or right answers. So there's definitely the thinking in that direction, but it's probably will be next year, because we are still developing what's the best from how to connect them to a chatbot to basically feed into and stuff. And in that case, is there's a developmental cost, so we need to do a lot of things before we launch that. That's a community thing. I realize that's not easy.
Definitely, features like that are so helpful to kind of bring the community together and kind of get an understanding of where the problems are, but then, you know, allow for other users to help other users. So looking forward to that feature next year. There's a question here on integrating multiomics with non-targeted MS data. What do you recommend in terms of integrating multiomics data with non-target MS data from chemical contaminants obtained from these samples, from the same samples, to assess association with exposure?
Yeah, this is Exposomics, and yeah, this is I think one of my example is integrating untargeted metabolomics with genes stuff, and the idea is that you do a pathway level integration. For example, we have this joint pathway analysis, and you can upload your genes or sequencing genes or whole stuff, and you can upload the pick list, basically this untargeted. Overall, that the exposure will hopefully perturb some pathways detected by transcriptomics, and perturb some pathway detected by metabolomics, and not necessarily they will agree. But if they are really in the diagonal part, they will agree with each other. If they don't, it's basically either the time difference, you didn't collect in stable state, or just like that. So integration of untargeted with other omics is, you can use this. Yeah.
Okay, great, thank you. There's a question here. When processing data in MetaboAnalyst, is it possible to take into consideration batch effect and apply a correction? If so, what algorithm would you consider or think about using? Or if maybe you already have something in MetaboAnalyst that can be, you know, used for batch effects in general?
Yeah, batch effect is critical, and a lot of batch effect is smaller. Into study, you can use some standard on normalization. You have common features, and potentially can help. If really just looking for a batch effect adjustment, and I think we have we have a feature in MetaboAnalyst. It's called other utils, and we have about 12 commonly used batch effect adjustment. One of them is Combat and ANCOVA, Wave ICA, and RS and AGAN MS. We have a huge batch effect. We saw that, and it's really, after we adjusted the batch effect, and all the IBD data became so meaningful. And they have control, and non-IBD is green, and they have Crohn's disease and red, and ulcerative colitis, and this is QC samples. So this one is only appearing after we adjust the batch effect. The original publication on Nature Biotech, I think, just do it as default. So they didn't realize the huge batch effect. Overall, that normalization using a common reference will help. And if that's not enough, you use the other utilities. So for example, here we have other utilities. You click and you will see a batch event adjustment. We have method provided. And in the end is you're going to have a things that really the slowest things give you distance among the QCs. The lowest one will be chosen. So sometimes you don't know which one is the best batch effect adjustment, and this one will help you automating the whole process.
Great, thank you, Dr. Xie. We have so many questions, but due to time, we won't be able to get through all of them. At this time, I'd like to share my screen to share some information about Bits and Bytes that was shared earlier by Oliver. And just to those new users, especially those new users, we do have a Bits and Bytes with Dr. Xie, and he'll be teaching Introduction to MetaboAnalysis, but it allows for any users to join to ask more of those questions that you may have or that we didn't get to today. I do want to point out that today is the last day for an early bird special. So again, Bits and Bytes is our short course series. We have 10 this year, and you can pick and choose, or you can take all 10 depending on your interest. These courses are really made for focusing on different topics and ideas in metabolomics. So if you are interested, please check out our website. I shared the link in the chat, but again, you can just Google that. And with that, I'd also like to talk about our next speaker. So in February, we'll have Dr. Lorraine Brennan from University of College Dublin. She'll be talking about metabolomics, a powerful tool in human nutrition research. She leads a metabolomics group really focused on nutritional research and precision nutrition. So we're looking forward to that and hope that everybody can join for that.
And Arpana, if you could maybe download them the questions and then send them simply the questions to Dr. Xie by email, maybe he can follow up later on the questions that we just didn't have time to answer.
There was many from new users, old users, you know, really thinking about the future of MetaboAnalyst and where it stands. So,
all right. So thanks everyone for joining, and we hope to see you soon again. And so many thanks to Professor Xie to entertain us and educate us about MetaboAnalyst 5.0 today.